Hello, and thank you for joining today's webinar, Translational Applications of MiniPDX, an in vivo organoid assay in new drug R&D, presented together with LIDE Biotech. The services and technology that LIDE will be sharing during this presentation are available for purchase on the Scientist.com marketplace. Simply search for MiniPDX or Shanghai LIDE, or request assistance from our concierge team. If you'd like to get in touch with the LIDE team, their contact will be available on the last slide. Now, I am pleased to introduce Dr. Danyi Wen, the speaker from LIDE Biotech. Dr. Wen's research foundation was 12 years at Millennium Pharmaceutical, Biogen IDEC in Boston, Massachusetts. In 2007, attracted by the innovation coming from overseas, she returned to China as a VP of Biology at Shanghai Chem Partner. She founded LIDE Biotech in 2011 to drive research and development in personalized precision medicine. Thank you for joining me today. You can now take it away, Dr. Wen.
Thank you, Megan, for your introduction. Very glad to have this opportunity with Scientist.com to introduce what we are doing at LIDE. LIDE actually founded about 10 years ago, and we are focused on the translational medicine. And we have two subsidiary companies. One is Xi'an LIDE, and the other one is Shanghai LIWEN. So both Shanghai LIDE and Xi'an LIDE has ALEC accredited animal facility, and LIWEN is a CAP certified clinical central lab. So LIDE is actually focused on the precision medicine. Does have two arms. One is for the medical services, that's a personalized oncology. The other part is supporting the new drug R&D. So on the personalized oncology part, and we know that at the clinic, only about 25% of the cancer have a clear cancer driver gene. For this kind of the cancer patient, just go to the NGS or PCR and those molecular level testing that can get the answer for the drug treatment. On the other hand, about 75% of the cancer without a clear cancer driver gene, then you have to go to more downstream, go to the cellular and also animal studies, so-called functional diagnosis. So LIDE on this part, we actually can offer both NGS services and also functional diagnosis. Then the other part on the drug R&D, and we using our translational platform, we actually can offer the pharmacology studies, and also with our drug resistant model, we were able to do some new drug discovery for the new target identification and validation work. And also at the translational stage, we are carrying out many investigator-initiated trial. So through those translational platform, we are hoping with this service platform can help clients to doing the new drug R&D. So this is my presentation outline. First, I'll talk about what's the functional diagnosis, so-called version 2 of precision medicine. So a couple of years ago, there's a review article actually stated that precision medicine for cancer should with next generation of a functional diagnosis. So-called functional diagnosis actually refer to in vitro and in vivo assays, and specifically is a conditional reprogramming and the Organoid Assay and also the PDX. So speak of the Organoid Assay, and one of the key opinion leader in the field is Dr. Hans Clevers from Holland, and he is also the board member of Roche. So last month, he actually visiting LIDE. So he has a lot of publication. In his review article, he also compared the pros cons of those conditional reprogramming and also Organoid Assay and the traditional PDX. So, and those according to the NCI website, they recommended 3 functional diagnoses. This including 2 in vitro assay in the cell-based assay and 1 in vivo. So in the in vitro one is the first one is a conditional reprogrammed cells. This is a early stage developed by the Georgetown University Richard Schlagel's lab. So by adding the ROCK inhibitor, the cells can be reprogrammed to the progenitor state stage. So gain the cell, gain the capability for the amplification, and use this one. People can get a small amount of the sample of the cells. They can be in vitro culture them. Then you put in the matrix gel. You also can grow into the 3D spheroid and organoid assay. And also, like traditional people doing the drug discovery, this has been using for 20 years. That's so-called patient-derived xenograft. It's a PDX model. So the golden standard is a PDX model for drug IND, but it takes a very long time. Usually need 4 to 6 months. So the in vitro assay and even is shorter, but the drawback is the way you giving the drug, you can only giving the once on the surface of those organoid. So in order to overcome those problem, we actually developed a so-called in vivo version of the organoid assay. We call this OncoV, a MiniPDX. Literally, this is 30 times faster than the traditional PDX. It is an organoid assay, but we actually put that into the mice, so we can give in the drug systemically for 7 days. And early this year, there's a review article on the cancer cells. They also described how get the patient sample, then through those functional testing together with omics, and they can give the patient the translational medicine services in about 4 weeks of the take-around time. And in our real practice, we actually, with the MiniPDX, we can shorten that to even just with 2 weeks. We can, together with the omics analysis, we can give the clinical physicians the answer. So the second part, I would like to introduce what is the MiniPDX, so-called the in vivo version of the organoid assay. So literally, we get the fresh patient sample either from the surgery or from the biopsy sample. Then we put into a mini capsule. So the mini capsule with a molecular weight cutoff of 500 kD of those kind of holes. So when we put in the cells into the capsule, we put that into the mice sub-Q. Then we're giving the drug systemically for 7 days. At the end of the study, we took out those capsule and using the cell titer glow to measure the ATP and on each samples. So we have do the many validation studies, and those are some real cases. For example, what showing on the picture is the patient is with endometrial stroma sarcoma. After first and second line treatment failed, and the patient was metastasized to lung. Doctor guided the biopsy sample, and we run the MiniPDX. We found out that the best responder is apatinib. Apatinib is actually angiogenesis drug. It's approved by the Hengrui Pharmaceutical for the second line failed gastric cancer. But in this patient, it is endometrial cancer, and based on this study, and the doctor gave the treatment with those apatinib, and those are the signs after treatment. So with those, and we have many of those cases. In this one, there's another case, and also like in the Shanghai Renji Hospital, they carry out the study. One group, they used the MiniPDX guided therapy, and the other one used the conventional therapy. And it turned out using a MiniPDX to guided therapy, the median overall survival and the disease-free survival can all extend more than 5 months longer. Then people are going to ask a question, what is the corresponding rate with clinic? So first we carry out some of the study with a fresh patient sample, and we found out at the clinic we got the sample, and we got the MiniPDX. At the same time, we also build the regular PDX. When we do the efficacy study, we get all the identical data that's the same as the clinical. So that's the left panel. There's 2 of those examples. Then we also using those kind of the PDX tumor. We're doing the MiniPDX assay to measure the so-called correspondence. Then, for example, in this case, this gastric cancer. At the clinical, when you using those ACE1 for no reason, and some patient is responder, some is no responder. So for the responder, we get also good result at the PDX models. Then we use the PDX tumor, we run the MiniPDX, we get the same identical result. So we know that the PDX correlate with clinical 89%. Then we published the methodology paper has been showing that MiniPDX, even that the shorter the duration to 7 days, but the correlate with traditional PDX 92%. In this way, by calculation, the MiniPDX actually correlate with clinic around 82%. In the real practice, we have collaborate with many doctors in the past few years. So far, already have a 12 ACI publication, and also have another publication on another 5 one is under submission. So they run on the different cancer type. The overall summary is overall response rate. MiniPDX correlate with clinic around 80%. So the third part, I like to introduce how we use those MiniPDX and omics analysis for doing the translational medicine. So first, like how people all familiar with the drug discovery process. During those process, there's 2 challenge questions. First is what's the clinical indication? Second is whether we can identify a biomarker that can use as the patient stratification. Then we want to start with a few cases. For example, we know the FGFR is a very hard target, and FGFR was identified about the same time with EGFR. EGFR soon become a drug, but FGFR, if you search the clinicaltrial.gov, they have been failed 310 trials. Many of those was run by the multinational big pharma. And so far, there's only 2 of those was approved. One is from Insight. They have been showing that under cholangiocarcinoma, FGFR2 fusion and rearrangement is the driver gene. But then you also see many cases. For example, AZ carried out a study. They used the FGFR2 amplification to enroll the gastric cancer, but at the end of the study group actually in compare with the control paclitaxel group, patient duration disease progress-free survival is 1.7 months shorter. So that apparently this is a failed clinical study. Because of those, that people saying that FGFR is actually a very so-called very complicated target. They have a multiple kind of gene alteration, either it's the mutation, overexpression, or translocation, or fusion. Which one is more like is the driver gene is not clear. So in this case, people come to us. We began to use so-called MiniPDX, running so-called basket trial or umbrella trial, trying to help a farmer to identify the clinical indication and also the biomarker. So when we're running on those functional testing, at the same time, people want to know what is the corresponding omics data. So like it's easier to carry out the genomic analysis on the DNA. But if you want to simultaneously doing the transcription level analysis, this part of the RNA analysis, you usually need a large piece of the tumor. So in order to save those issues, we also developed 2 assay kit. We call those K-cell RNA-seq and K-cell DNA-seq. With those, we only use a couple thousand of the cells. We can run whole exon sequencing and the whole RNA-seq. And also at the same time, we developed the so-called proteomics technology running the imaging mass cytometry. So in next few slides, I want to go to a little bit more detail about those. So because we're using the small amount of the sample to doing the analysis, so first we need to doing the validation. So we compared those the RNA sample and also DNA sample from either the bulk tissue or with just a couple of thousand cells, and we were able to showing that even is with a very small amount of the cells, we can get about the same coverage rate as the bulk tissue. And after those, then we also facing challenges when we getting the transcription level of the data. Usually, it's people look at on those heat map. Then people also need more kind of advanced tool to do the analysis. So we kind of developed a so-called function genomic image analysis tool. So in those and at the kind of bioinformatics level, and we were able to group that into the different subgroup category. So by DNA sequencing, we can get the information for the target therapy, but in combination with those transcription level analysis, and we do the FGI analysis, we also can know the microenvironment of immune therapy and also angiogenesis and the chemotherapy and also target therapy. Then those are a few examples. So each of the tumor sample, we're going to have those so-called cloud map. And if like GTAX Z-score larger than 2 is upregulated, showing as the red color, and if downregulated, going to show as the blue color. So if it's showing the proliferation signal is high, that means this patient potentially going to be sensitive to the chemo drug. And if double click on those genes, you also can block that on the wiki pathway and the reactant pathway. And the same of those, and if we identify the molecule that has the pathway activate, in this case is the PIK3, then we use the Roche PIK3 inhibitor. We also showing the good validation. And even interesting is actually is the Merck actually running a study. They carry out the study with all the Keytruda and using 220 patients covered 9 different cancer type. So all the Keytruda responder, non-responder, they did the transcription level analysis, and they published the result at the JCI, and they showing that around 18 markers in this study. They showing that as the responder for the Keytruda. And when you do the analysis, so among those 18 genes, many of those is the T-cell related, and also including so-called interferon gamma stimulated gene called ISG. So with those, and also with some other studies, we developed a signature called the interferon stimulated gene. And if it's upregulated, potential showing the patient going to be the responder to the those kind of IO drug like Keytruda. And if it's the downregulated, it's the no responder. So we have a comprehensive scoring system to running this. And we have been talking the DNA analysis and also the RNA analysis. And another important component we cannot missing is the so-called proteomics analysis. We introduced the technology called imaging mass cytometry. Literally, this is the technology developed several years ago at the state, and the professor they developed using the heavy metal to label the antibody, and then they use the CyTOF to doing the reading. And after those, and they have been so far have a totally 14 color heavy metals. So in this case, you can do the flow in with 40 totally different colors. Later on, they developed new technologies, this instrument called HAPRI. So then they can use those paraffin block slides simultaneously staining 35 of those markers. So in this way, you not only know which protein was upregulated or downregulated, you also know their location. You know that is on what kind of the cancer type. So we developed those 35 kind of the marker ready to go. And for this kind of the single molecule protein analysis, you also can do in the cluster analysis and to do the grouping. So those are the ready to go different panel have been developed. So based on the customer need, we also can customer design on those 35 panels. So after those, I've been talking about like how we use the functional assay together with the omics analysis to supporting the drug discovery. Next, I want to share a real case study. In this case, is a biotech company. They developed the drug. So first, they run the CDX model, found out in the type A tumor is very efficient. Then through the kind of the literature search, they notice and some like clinical study carried on at the state, they also including the other 3 cancer type. So they designed the clinical study at the phase 1 and the 2A. They including the 4 cancer type. The problem is after those phase 1 study, they didn't find which cancer type have a very good response rate, especially for the type A. They put a lot of hope, but during the phase 1 study and the /2A, they don't really see a very good responder. So they found us, then they using our MiniPDX. They said that we want to get a fresh patient sample with those 4 kind of the cancer type. Then we run the MiniPDX together with omics analysis. We want to see if we can, through this approach, we can identify the clinical indication. So the study result is the first round, they run around 31 samples. So only 5 responder. Overall responder rate is only 16%. Then they do the more detailed analysis. They found out that their best bet on the type A is actually on the 7 samples, we don't find only single responder. So this is the same as their clinical study result. Then they found out the type B have a much higher responder rate. So they changed the study design. They asked us to including more of those type B of the tumor type to doing the MiniPDX. So around those, when we run 20 samples, we found the 7 responder. So even without the further characterization, by selecting the right cancer type, the overall response rate already increased to 35%. Next step, then we give those responder and no responder. We run on the K-cell analysis. So we get the result. We found out if it's the responder in the common substrate, there's a specific mutation. So by this kind of the approach, and those biotech company, through those preclinical study, they not only identify the clinical indication, they also found out the corresponding molecular marker. So these days, more and more biotech company, even their drug already in the clinical stage, usually like a leading PI, they send them back to running our MiniPDX mouse trial to get the clinical indication and the corresponding biomarker. So from that, I've been introduced how we use the MiniPDX doing the supporting the drug discovery. Now I continue my presentation. So the first 3 topics we talk about how we use the functional assay, so-called MiniPDX, together with omics supporting the drug discovery. When we're doing the MiniPDX, simultaneously we also build the regular PDX. So and in the next 2 topics, and I'm going to further introduce how we're using those regular PDX to supporting the drug R&D. So far, far, we already established 1,500 of those PDX, and many of those actually is harbored a very specific genetic alteration, and also many of those is the patient already developed drug resistant. So the table listing here is showing the different PDX model that's resistant to the different drug, and also with a specific gene alteration. So if any people interested in those model, feel free to contact us. Then we also carry out a study with physicians. In this kind of the study, we got a group of the PDX model. Then doctor gave us the treatment they have been using at the clinic. Then we're using the same drug. We run on the efficacy on the PDX model. The conclusion is the PDX result almost 100% identical with their clinical outcome. Then we also want to introduce when people develop those drugs, they usually first go to the liquid tumor. In this case, we actually developed those so-called orthotopic model, and we get the fresh patient bone marrow sample or the peripheral blood sample. We put in the femur of the mice, and after those, we use the flow cytometer to monitor whether we can successfully build the model. After we establish the model, we actually take the spleen and frozen them down. Later on, when we need to redo the efficacy study, we take the spleen, we re-put that back into the femur. In this way, we can run those liquid tumor orthotopic model. So the example in here is one of the client running the CD47. So we use those AML model and successfully running the efficacy study. So the left panel of those table and showing that all the PDX model we have been established so far on the liquid tumor, including the AML and the ALL, and also all kind of the CLL and transform to the diffuse, the large B-cell lymphoma, and also some multiple myeloma, etc. And also, at the clinical, one of the challenges at the late stage, many of those tumor metastasize to lung. So with those, we also established technology to doing the orthotopic model of the brain tumor. So in this case, we generated those PDX matching cell line. We also used the luciferase doing the labeling. Then we used the stereotoxic apparatus to doing the injection, use the IVS imaging. So in this case, we have been using the glioblastoma model. We also have all kind of the tumor, for example, the breast cancer and also the gastric cancer, HCC or non-small cell lung cancer, those metastasize to lung. So if people interested to develop the drug to overcome those metastases, and those the model we can offer to. And at the same time, we also have a very extensive effort on those so-called IO models. So most popular used is using the human PBMC to doing humanize the mouse model. And those kind of things we can all together with the cell line, and also we can doing that with the PDX. And put at the bottom is the list is the here. We all successfully established and also using the data for the IND. So because all the donor we have been pre-screened, and the clients running the study with us on those kind of the model, we can get very consistent repeatable result. So if people are interested in working on the IO model, hopefully in the early next year, we're going to have another webinar specialized on those IO models. Feel free to contact us. So then I want to making the summary is traditional doing the drug discovery. People use those genetic engineered cell lines. Then they go to those cell line derived CDX model. But this model correlate with clinical less than 5%. What LIDE offering is using those translational platform. For example, in the cell-based assay, we can use those conditional reprogrammed cells. And also also during the screening cascade between the cell-based assay and the in vivo model, we also have those MiniPDX plus omics analysis used as the screening cascade for the faster turnaround assay. Then we also can go to the regular PDX model and come to the clinical stage, the OE, like a pre-IND stage. And many biotech companies facing the challenges for identify the clinical indication and also for the patient stratification. In those cases, they actually join our MiniPDX mouse trial using the fresh patient sample for clinical indication identification, and also in combination with omics analysis, they try and identify the biomarker. So this is how we're using the functional diagnosis to support the new drug IND. Then the last one I want to talk about, since we have many drug resistant model and we want to use that to identify some new target to overcome those drug resistant. So what we do is we get a fresh patient sample, and either we can build the PDX model, or we also can use the fresh patient sample to directly using the conditional reprogramming, building those cell lines. And if we build the PDX model, we also can use the PDX model as the starting material to build those cell lines. Once we get those cell lines, we isolated the RNA, and we using the pro-ACI RNA technology to do the validation, to doing the screen. And after first and primary and the secondary screen, and we also use the CRISPR and the control compound to doing the second round of the target validation. So when we get those hit potential, it could be like it's a new target. In those target, in combination with bioinformatic analysis, we can try to identify the new hit for the small molecule and also for the large molecule. We also could identify some known target that is more like a repositioning of your drug candidate to the different indication. So those are first we took the example is we have a very unique model. This patient over the year of the treatment, they developed the drug resistance. The patient carried on the exon 19 deletion and also have the T7090 mutation. After used those ositinib, they also developed the third mutation, is a C7097S mutation. So by now, maybe this is the only one in the whole world naturally occurred on the patient, is a triple mutation. Because the unique of those model, we have been using this model to support many multinational big pharma to running their drug screen. And then we also use the conditional reprogram technology. We were able to building those matching cell line. So when we're building the cell line and we're doing the TCA, we found out this the PDX model is resistant to the ositinib, or called AZD9291 from AstraZeneca. And when we're running the cell-based assay with those same matching cell line, we get the same result. The IC50 is very insensitive, is 2.01. Then the other cancer, the cell, the PDX tumor is actually sensitive to those ositinib. When we're building the cell line, when we're doing the study, we get the same identical result. The IC50 only 0.03, so it's also sensitive to those drugs. And we also build those so-called wild type and without the EGFR mutation. Now we give those ositinib, and those also you showing is the resistant. So what we got those cell line data is corresponding with the PDX data very well. Then after confirmation with those cells, we isolated the RNA. We're also using a so-called pro-RNA technology. And we're doing the dose screen and identify through the primary screen and the secondary screen, we identified a whole bunch of those novel hit. Through those kind of the target, and some of those, we also found that there's a two compound. So we used the two compound, we did the validation, mutation, and some of that we validated with the CRISPR. So through those, we also can offer the target identification and the target validation work. So far we have been served more than 200 clients and over 1,000 of the project. So many is overseas multinational big pharma and the biotech, and also we have many domestic companies. So putting together, hope LIDE's translational platform can help those pharmaceutical company and also biotech to on their drug discovery process. So if you have any further questions, feel free to contact our executive team at the US. So the contact person is Josh. His contact person is listed on this slide. Thank you for your attention, and I close my presentation here.
Thank you, Dr. Wen. That concludes the presentation portion of this event, and thank you to the audience for following along. I hope this has given you some insights into some of the services available through LIDE. We can now get to some of the questions that you all have asked. So the first question for Dr. Wen is, with a 92% MiniPDX compared to traditional PDX, why would organizations choose traditional, and what's the benefit to that?
Yeah. Thank you for the question. So MiniPDX is only 7 days, so it's more used to use like rank order your drug. And traditional PDX, that's going to be systemic dosing for 21 days. So that can give you a more accurate IC50. So when we're doing the drug discovery, and most people using the MiniPDX as the screening cascade between the cellular assay and the regular PDX. So they use that MiniPDX to get a quick answer, which one is a best responder. Then they use the regular PDX to get the accurate IC50, and for the EC50, then they use those for the IND filing.
Thank you. And what animals and strain are used for the PDX and the MiniPDX model?
Yeah. So the PDX model, and that's usually we use the nude mice or skid mice, and the MiniPDX we usually use the NuNu mice. So that's depend upon the model, and if we use the immuno-oncology model, then we use those NSG mice. Those are IL-2 receptor gamma chain deficient. So depend upon the model, which model we use. We maybe use a different strain.
Thank you. And what is the approximate timeline for an in vivo PDX study?
So the traditional PDX study, and if the tumor currently is warm, then it's take about like 2 months, because your dosing going to be need 21 days. But if the tumor is frozen, and that's going to take longer, because we need some time to recover the tumor first. And then it goes to the regular PDX model. The turnaround time is only 7 days, so it's faster.
Excellent, thank you. How many passages did you use for PDX?
We actually did a systemic validation with a multinational big pharma, and they did a different passenger, and they found out after passenger 10, and those model lost the so-called heterogeneity. It's more like a CDX. That's why like people prefer to use the model for the efficacy study within the passenger 6, and the maximum shouldn't be over the 10 passenger. So majority of our efficacy study done at LIDE in between the passenger 3 to passenger 6.
Thank you. Another question for you: how can we access capsules for MiniPDX?
Yeah, that's a good question. Currently, and we have those so-called RUO, the research use only kit. We are in progress of get those registration as IVD. So if people want to acquire that, and we can providing those as the RUO kit. but it's not registered IVD yet. That's in progress.
Thank you. How do you ensure that a MiniPDX is growing after 1 week and that you are not treating dying cells?
Yeah. So when we're doing those MiniPDX, we have for each treatment, we implemented the cells into 6 different capsules and implemented into 2 mice. So the end of the study and each point is going to be n equal to 6. Now we have the control group without any treatment between the day 1 and also the day 7. And then we do 2 times the measurement. So that's window going to become the 100%. Then all the treatment group are going to be in compare with this control group. So if the cells do not grow, and that's mean it's going to be so-called a failed study. But in our experience, since we put those so-called organoid into the mice, so the cells doubling time is much better than we grow in vitro. Majority of the case, and those primary cell, they all can grow over 2-fold, and many of them even to 5 to even 10-fold. So they can the case like Julia described here, and if it do not grow, and we're going to consider that as the failed study, but that percentage of this is very low.
Excellent, thank you. Another question for you. Do you think 3D cell culture can replace PDX?
I don't think so. So the 3D cell culture, and also we have that and the so-called conditional reprogramming, and also the traditional organoid assay, we all have that. Then we also developed the so-called in-vivo version of the organoid, is the MiniPDX. That's the presentation of today. So each of those assays have the pros and the cons. So for the 3D culture, when we're doing that in vitro, the major drawback is the way you administer the drug is you can only give the ones from the surface of the spheroid. And actually our drug was given systemically. That's why by putting those organoid in vivo, and we were able to giving the drug systemically in the 7 days. That's why for the traditional 3D culture, you can test the chemo drug. Also, majority paper is published all testing the chemo drug. But when we're running the MiniPDX, when we put in the in vivo, we can running the test both the chemo drug, we also can test those angiogenesis drug, and also test those pro-drug and the ADC and the PROTAC, so, and also target therapy, so it has much wider application than the in vitro assays.
Thank you so much. Can you tell us the difference between MiniPDX, regular PDX, and organoids? So the organoid, as I mentioned, is more like a 3D sphere with some matrigel. So that's a culture in vitro. So the way is that you're giving the drug, you can only giving the ones on the surface. And then we put that into the mice, so-called in vivo version. So MiniPDX can systemically giving the drug for 7 days. But even for those, it's not the real growing in the tumor. So the traditional PDX is still the golden standard. When people filing the IND, in order to get the EC50, they still run the traditional PDX. So they run those cell-based assay, 3D culture, then they go to MiniPDX, then they find the best one to do the regular PDX. So those are the application scenario was different, so they complement each other. And what kind of drugs can the MiniPDX test?
Yeah. So the MiniPDX can test on those angiogenesis drug, and also the targeted drug, and say those pro-drug like ACE1 in the gastric cancer, and also we test a lot of ADC drug and the PROTAC drug. And so that's a current version. We do not test the the IO drug, because for the immuno assay that need doing the PBMC reconstitution. Currently, we're running that in the regular PDX or with the CDX, but not on this MiniPDX version. So MiniPDX can test all the drug except for the IO drug.
Excellent. And it looks like we have just one more question for you. What is the advantage of K-cell DNA-seq and K-cell RNA-seq?
Thank you. So when we're doing the functional assay, naturally people want to understanding why some is a responder, some is no responder. For those, then you naturally need to go to the genomic analysis. But with the traditional way, when the people doing the transcription level analysis, they're going to need a big chunk of the tissue. That's why we developed those so-called K-cell system. When we prepare the cell for the MiniPDX, some of the leftover cells, couple thousand of cells, we can run the whole exon sequencing for DNA, and also we can run the whole RNA-seq. So in this way, we can get the result from same tissue, from the functional assay. So that can give you the good correlation and also can give you the explanation in the molecular level.
Excellent. And it looks like we had a couple more questions just come in. Do you label the primary cells in the capsule?
No, we don't. When we do the isotopic model, we label that. When we're doing those MiniPDX, usually is used a fresh patient sample. So we just freshly prepare that and directly run in the assay. We do not label that.
Okay. And can you tell us how to build up the pro-siRNA library?
Yeah, and that's we maybe can schedule a meeting to talk that separately. And basically, there's a professor, and they have the patented technology by co-expression of P19 protein, and they actually can express the RNA in the E. coli system. So made that screening very high throughput and low cost. So that's how we're using this system for the primary assay, for the target identification, for the drug-resistant genes. So if you are interested in the detail, and you can contact Josh, and we can schedule a TC to talk about in a separate.
Okay. And how can you run a target identification with your drug-resistant model?
Yeah, so we have those drug-resistant model, and for example, is ositinib-resistant non-small cell lung cancer. Then we use the conditional reprogramming. We build the matching cell line. After those, then we use those pro-RNA technology to screen what is the important gene, and then in combination with the bioinformatic information for the analysis of the drug sensitive and versus drug responses. Then we also identify some interesting target. Then we also use the CRISPR technology and to do the second round of the validation. For some of those targets we identified that already have a 2 compound available, so we also use the 2 compound to do the validation. So with those, we identify some many interested target, and in progress of trying to get some kind of hit from those drug-resistant model. So far we have over 100 of those cell lines, and we systemic getting all the targeted drug. Then we establish the PDX and also have the matching cell line. So any people interested to get the some new target to overcome those drug resistant, and I think this is a platform can offer the unique value.
Excellent. Thank you so much, Dr. Wen, and thank you all for joining us for today's presentation.
Thank you, Megan, and thank you for all the audience.